Hello, team. This is Dr. Chris Alnagallo, Assistant Professor of Science with Chamberlain University, Phoenix campus. In this video, we will address key concepts in the upcoming celebration of learning, more formally known as exam number 2. While this presentation should not be considered a comprehensive review guide, it highlights important concepts. And by important concepts, I mean these concepts are high yield, and it is imperative that we understand how these concepts work and operate so that we can apply this knowledge base to various applications in anatomy and physiology. The upcoming celebration of learning number 2 will cover material that was addressed in weeks 3, 4, and a little bit of 5. The slides that follow are borrowed from the PowerPoint presentations associated with your textbook. So we begin with cellular form and function, which we addressed in week 3. First and foremost, the basic unit of life is the cell. You need at least one cell to be considered life. So cells will need to have a boundary, a clear demarcation that marks who is a part of the cell and who is not a part of the cell. This structure is known as the plasma membrane. It is also referred to as the cell membrane. It is comprised of primarily lipids, specifically phospholipids, which have a hydrophobic, so a water-fearing area, as well as a hydrophilic, meaning a water-loving area, and several proteins. More on the proteins later on. Any molecule that has a hydrophobic and hydrophilic side within the same molecule is known as an amphipathic molecule. Amphi as in amphibian, and amphibians can be in both land and sea. So that's something that I used to remember what an amphipathic molecule is. Now, contents inside the cell, meaning they are within the confines of the cell membrane. This area is known as the cytoplasm, and the cytoplasm will be comprised of fluid, just like the outside of the cell is comprised of fluid. This fluid is the universal solvent, which we know as water. Water will have different types, different names based on its relationship, its location to the cell membrane. If this water-based fluid is outside the cell membrane, then it is considered the extracellular fluid. If it is found inside the cell, then it is known as intra, meaning within cellular, within the cell fluid, often abbreviated ECF and ICF respectively. Now, the intracellular fluid is also known as the cytosol, which is not necessarily synonymous as cytoplasm. So the cytoplasm comprises of the cytosol, as well as the many compartmentalized areas of the cell, each designed to perform a specific physiological function. More on that later on. But in addition to the cytosol and organelles, the cytoplasm also includes the cytoskeleton. And the cytoskeleton is comprised of several proteins that form the framework of the cell. It gives the cell a bit of shape. So once again, the cytoplasm is not necessarily the same thing as the cytosol. Here is a very useful schematic diagram of the cell. Highlighted in yellow are organelles that are really important in the daily functioning of the cell. So first and foremost, at the center is a nucleus. The nucleus is a really important organelle. It is designed to house the genetic material of the cell. This is also known as deoxyribonucleic acid, often abbreviated DNA. DNA is really important. It basically provides the genetic makeup of the cell, and so it cannot afford to be damaged by even the proteins in the form of enzymes that are found in the cytoplasm. For this reason, the genetic material, DNA, which has a genetic code, will be housed in the nucleus, which has a nuclear envelope also comprised of a double membrane. There's a specific area within the nucleus located here in darker purple. This is known as the nucleolus. In the nucleolus, ribosomes are made, and ribosomes are the other organelle that is worth paying attention to. There are free ribosomes, these dotted structures here, that are also found dispersed around the rough endoplasmic reticulum. Ribosomes are comprised of ribosomal RNA, and they are made within the nucleolus. Now, the maze that is found around the nucleus is known as the endoplasmic reticulum. There are two parts to the endoplasmic reticulum. There is a rough endoplasmic reticulum and smooth endoplasmic reticulum. The best way to identify the difference is to look for the presence of ribosomes. Ribosomes will only be found in the rough endoplasmic reticulum. And if you look really closely, the rough endoplasmic reticulum will have these dots interspersed throughout it. And because of this, they create a rough structure, a rough looking texture, if you will. These dots are ribosomes. As the endoplasmic reticulum moves away from the nucleus, we lose the ribosomes. And because of this, the texture of this area is smooth. And for this reason, we name this area the smooth endoplasmic reticulum. The ribosomes are really important for protein synthesis. They are key for a phase of protein synthesis known as protein translation. And any protein that's being formed will eventually make its way to another organelle that is also membrane-based. What I'm referring to is the Golgi complex, also known as the Golgi apparatus. Here, tertiary and quaternary structure of protein will finalize, and they get packaged into these vesicles, which essentially carry these proteins to the surface of the cell so that they can be secreted and so they can travel to different parts of the body. The vesicles are also lined with a membrane-based lining. So hopefully you're seeing a pattern here, and that is many organelles that are involved in protein synthesis will have a membrane-based lining. This is because proteins do not easily pass through the phospholipid bilayer that is the cell membrane. And so the membrane comprised of a phospholipid bilayer can easily fuse and separate from the endoplasmic reticulum Golgi complex and Golgi vesicles to move protein throughout the cytoplasm of the cell. Another organelle worth mentioning is the lysosome. The lysosomes are signified by these darker blue-purple structures. There are several of them throughout the cell. They are tasked with destroying foreign material or recycling contents, macromolecules that the cell no longer needs. In many ways, the lysosome is the recycling depot of the cell. If the lysosomes are the recycling depot of the cell, then the mitochondria, which the cell will have many, is the powerhouse of the cell because it makes a nucleic acid that is energy packed. This nucleic acid is a 3-lettered abbreviation, ATP, which stands for adenosine triphosphate. So the mitochondria are really important for a process that drives metabolism, and this is aerobic respiration. Aerobic respiration requires the presence of oxygen. Anaerobic respiration has limited supply of oxygen. Generally, aerobic respiration will produce way more ATP than anaerobic respiration. So in aerobic respiration, the main product, the whole reason for why mitochondria will be involved in converting a glucose molecule and water is to make ATP. In the process, it makes 2 byproducts. These are carbon dioxide, which will then be exhaled from the lungs, and water. Now, water is a byproduct. We don't perform aerobic respiration to make water. So it, as a byproduct, contributes to extracellular fluid or intracellular fluid, which is water-based. The last highlighted organelle on the slide are the centrioles. The centrioles will appear and disappear based on whether the cell is preparing to undergo cell division or not. There are many other organelles in the cell. If they are highlighted on this slide, I highly recommend paying close attention to these organelles, be able to identify them, and be able to identify what their main roles are. We are now going to hone in on the plasma membrane. So the plasma membrane is a phospholipid bilayer, 2 layers of phospholipid molecules. There are 2 types of proteins located on this plasma membrane. There are these transmembrane proteins and peripheral proteins. Transmembrane proteins and peripheral proteins are simply a reflection of the location of these proteins with respect to the plasma membrane. If the protein spans the entire width of the phospholipid bilayer, then we are looking at a transmembrane protein. If it is only found on one side of the phospholipid bilayer, and this will typically be on the cytoplasmic side, the side that's facing the cytosol, such as in this example here that I'm pointing out with my cursor, this is known as a peripheral protein. Again, the terms peripheral and transmembrane protein simply refer to the location of the protein with respect to the plasma membrane. This image is an overview of various transmembrane proteins. We know they are transmembrane proteins because they all span the entire width of the phospholipid bilayer, but there are different functions. In green here is a receptor. A receptor is designed to instill changes, physiological changes inside the cell in response to a signal such as a chemical messenger, represented by this red triangle. There is specificity in the chemical messenger. So there is a lock and key model, so to speak. Each chemical messenger has a receptor that's designed specifically for it. And this allows for organization and synchrony in cell processes. This means this receptor will only activate processes within the cell if it binds, if it meets the specific chemical messenger it is designed for. Receptors are a really important way for the cell to communicate with what's outside of the cell. So in many ways, the plasma membrane is like a wall. It is a boundary that is selectively permeable. So it is designed to keep things out that should stay out of the cell and keep things in that should stay within the confines of the cell. But it also allows for certain molecules to move into the cell when the time is right, or certain proteins and other signal factors to leave the cell if the time is right. Another transmembrane protein I'm going to bring to your attention is one that's represented in light blue. This is an enzyme. We've talked about enzymes already. An enzyme is a catalyst. It is a catalyst, meaning it speeds up the rate of reaction by decreasing the activation energy. So an enzyme will speed up the chemical reaction rate. A channel is also a transmembrane protein. It allows for certain molecules that may have a hard time passing through the phospholipid bilayer, not because it's not allowed, but because of its chemical structure. So remember, the lipids in the phospholipid bilayer don't like to be around other molecules that are not lipid-based. They don't do well with molecules that are charged or molecules that have a large size. And so for this reason, any molecules, specifically small molecules that have an overall charge, which we call an ion, if they need to get into the cell or if they need to leave the cell, they will find a channel which has a tunnel at the center that allows for safe passaging of these ions into the cell or out of the cell. This shields them, this channel shields them, this overall charge that is an ion, from the hydrophobic area of the phospholipid bilayer. Other transmembrane proteins serve as cell identity markers because of the presence of a glycoprotein, a sugar protein molecule that acts like a flag. And then there are transmembrane proteins that are responsible for keeping cells together. We have pictured here a cell adhesion molecule, often abbreviated CAM. We can see how this is just half of the bigger cell adhesion molecule. It is anchoring these two cells to one another. We're going to hone in on a concept known as the second messenger system. Here we bring to the forefront two transmembrane proteins we have already identified, and that's the receptor and the enzyme. Recall that all enzymes have a last name that ends with ASE. So here is adenylyl cyclase, and looking at the second half, the ending of the last word or last phrase to name an enzyme, we know we're looking at an enzyme if the last three letters are ASE. Another example of an enzyme is kinase, which we will talk about a little bit later. So the whole goal of the second messenger system is to allow for the cell to perform some sort of function in response to certain factors that the cell receives from the outside, the extracellular fluid, without that signal passing through the cell membrane and into the cell. That is where the receptor comes in. So the receptor responds or binds to the first messenger. The first messenger is any chemical that stays outside the cell. It is the first messenger because it is the first to communicate that there is a need for some sort of cellular process to ramp up within the cell. Binding of the first messenger to its respective receptor is the first step in the second messenger system. When there is binding of the first messenger to the receptor, this transmembrane protein that is a receptor undergoes a conformational change. It shifts the folding of the protein such that it will release G protein, a peripheral protein, which will allow it to communicate to adenylyl cyclase, which is a transmembrane protein enzyme. Now, G protein is not a messenger. It is there to group, to couple this enzyme with this receptor. Without this coupling system, adenylyl cyclase will not be associated with anything that happens at its respective receptor. When G protein makes contact with the enzyme, that activates the enzyme, and it activates the enzyme. What it does is it will break down ATP into two phosphate groups, because remember, energy and matter is never broken or made. It's only transferred. This is the law of conservation of matter, which you'll talk more about in chemistry. The bottom line here is we should be able to account for every phosphate group that was released from ATP. We denote a phosphate group by the capital letter P and subscript I as such here. So when ATP is broken down by adenylyl cyclase, it makes two phosphate groups that will have some use later on for the kinases. that we'll talk about a little bit later. But it also leaves cyclic AMP. This is the third phosphate group. So there was no phosphate group that magically disappeared. The last phosphate group stays bound to the adenosine. That's the A in ATP, as well as the A in AMP. Now we have AMP, adenosine monophosphate, because there's only one phosphate group remaining. Cyclic AMP is a rounded molecule. And in this form, cyclic AMP, it has the capacity for amplification. Cyclic AMP is the second messenger. So the first messenger never entered the cell. The second messenger, cyclic AMP, was made within the cell in response to the first messenger. Cyclic AMP is responsible for activating a group or a class of enzymes known as kinases. And kinases are generally responsible for moving phosphate groups to make ATP or to break down ATP. Amplification is a concept that we associate with cyclic AMP because it is like the CEO of an organization. One cyclic AMP has the capacity to activate many kinases at one time. So, in the CEO analogy, when the CEO wants to initiate some sort of project, he or she simply informs the vice presidents, who will then give direction to all the managers, who will then give direction to all the team leaders, who will then make sure all hands are on deck to make sure the end goal is achieved. One cyclic AMP has the capacity to activate many enzymes, including kinases. This is the second messenger system. What we will move into now is a discussion on how certain molecules, including water, can move into the cell and out of the cell. Before we move into water, let's address the respiratory gases. The respiratory gases are oxygen and carbon dioxide. We breathe oxygen in from the external environment so that the circulatory system, comprised of blood vessels and blood. Here we have pictured at the center a red blood cell. Red blood cells responsible for carrying oxygen to different parts of the body and carrying carbon dioxide, which again was metabolic waste. It was a byproduct of aerobic respiration we talked about earlier. Carbon dioxide, a small percentage of carbon dioxide that is released from the cell as metabolic waste travels through the blood vessels to return to the lungs so that we can exhale it. Now, because these are respiratory gases, they move into and out of the cell by a process known as simple diffusion. This brings us to the definition of diffusion, a concept that will be applied in many scenarios, and so therefore really important to understand. Diffusion is the net movement of particles from an area of high concentration to an area of low concentration. This is a natural law of the universe. movement of particles from high concentration to low concentration. This is what we mean when substances move down the concentration gradient. They're moving from an area of high concentration to an area of low concentration. An example that I use in the classroom setting of diffusion is that of perfume. If I were to hold that perfume bottle right next to me at the front of the classroom and sprayed 2 or 3 blood vessels, let me backtrack a couple of seconds there. So I was explaining using an analogy, diffusion. If I held a bottle of perfume at the front of the classroom and applied 2 pumps, within the first second, I, at the front of the classroom, would smell the perfume. But my students sitting at the back of the classroom would not have smelt that perfume within the first second. They may smell the perfume after 5 minutes, and that's because of diffusion. The area at the front where the perfume bottle was sprayed released perfume particles, which explains the high concentration area that was the front of the classroom. At the back of the classroom, there were no perfume particles, so this was an area of lower concentration. Diffusion dictates that particles were moved from an area of high concentration, the front of the classroom, to an area of low concentration, the back of the classroom. This is known as simple diffusion. It does not require a membrane, and respiratory gases, oxygen and carbon dioxide, move into and out of the cell using simple diffusion. This is the simplest case of movement of particles across that selectively permeable membrane that is the plasma membrane. In contrast to diffusion, there is active transport. Active transport requires going against the concentration gradient, meaning particles are moving from areas of low concentration to areas of high concentration. And so because of this, we need ATP. We need to put in energy into the reverse process. We need to put in energy anytime we try and move particles from areas of low concentration to areas of high concentration. If we use a hill as an analogy, the top of the hill would be synonymous with high concentration, and the bottom of the hill would be synonymous with low concentration. It is easier to run down the hill because the force of gravity is typically helping you. This represents how easy it is for particles to move from areas of high concentration to areas of low concentration. Now, what would happen if one were to start at the bottom of the hill and have to climb that hill? Many will say this is more difficult or more challenging than moving down a hill, but it is representative of how energy is required, effort is required to move particles from areas of low concentration to areas of high concentration. More on that later on. Anytime we follow the diffusion of water across a selectively permeable membrane, this process is known as osmosis. And that is really important to make sure that we are following the right particle, either the solute or the solvent, based on what the question is asking of us. It is very important to understand that osmosis requires following the solute. Typically, this is water. Let me backtrack. Solvent. Osmosis requires tracking the movement of solvent. This is typically water because the universal solvent is water. When we see terms like tonicity, we are referring to a state of concentration with reference to the solute, Typically, solutes and solvents will have an inverse relationship. If the number of solute particles increases, then effectively the number of solvent particles have decreased. Osmosis is really important for helping us understand why we can't just give patients sterile water. It has to do with the way fluid, water-based fluid, will redistribute themselves or itself on either side of the cell membrane. In this schematic diagram, we illustrate osmosis. So in this beaker, this perforated line at the center represents a selectively permeable membrane, much like the plasma membrane that surrounds the cell. In light purple are particles that represent the solvent, in this case water, and in larger red circles are the solutes. So there is an inverse relationship between solute and solvent. So in side B of the selectively permeable membrane, there is more solute particles when compared to solvent particles. And so the solvent particles will be present in a lower concentration inside B compared to what we find inside A. So let's get the lay of the land here. In a beaker, before any osmosis, there's an area of high concentration of solvent, aka water, on side A of the beaker, and lower concentration of solvent, water, inside B. Because osmosis is the diffusion of water, diffusion meaning movement from areas of high concentration to areas of low concentration. After quite some time, 30 minutes later to be specific, there will be more water particles, more solvent particles on side B when compared to side A because of the process of osmosis. For this reason, we cannot give a patient sterile water. There is a common misconception that because water is the universal solvent, well then, you should give the patient as much water as possible. That is theoretically true, except for we have to evaluate the effectivity, the possibility of osmosis, which will redistribute water in different places. And so more water is not necessarily the answer. The right concentration of water may be the answer. And so we will move now into the effects of tonicity on red blood cells. Red blood cells are responsible for carrying oxygen throughout the entire body. For this reason, red blood cells are critical. They are very important to the body's ability to thrive, not just survive, but thrive. Red blood cells will travel in blood, and while that fact is, one may argue, common sense, what may not be intuitive is how different concentrations of water, when infused into the bloodstream by, let's say, an intravenous route, an IV, how the concentration of water can affect the shape of the red blood cell. We ideally want red blood cells to have a biconcave shape. What this means is it is thicker around the edges than it is at the center. This biconcave shape allows for red blood cells to travel through different size blood vessels in the circulatory system. They will allow red blood cells to fold and bend as necessary to reach the smallest blood vessels of the body so that gas exchange can occur, so that the cells can get access to the oxygen that these red blood cells are bringing to them. At the center is an example of an isotonic solution. This is what the red blood cell looks like when an isotonic solution in the form of an IV is infused to a patient. This is what we want because this is what will keep the shape of the red blood cell a biconcave shape. If the concentration of water is too much or too little, because of osmosis, it will change the shape of the red blood cell. You may either have a red blood cell that swells up so much because osmosis is driving water from the outside of the cell and into the cell. This can essentially burst the cell, and bursting the cell will effectively kill the cell, rendering it useless to the body. In the other extreme scenario, the cell shrinks in the process called crenation. Here, water leaves the red blood cell for the extracellular environment, leading to the shrinkage of this red blood cell. We neither want what's happening in scenario A or what's happening in scenario C to occur if we can help it. What we want is the red blood cell is at the center of the slide here, pictured as figure B. What type of solutions will result in different red blood cell shapes? So we're going to take the simplest scenario at the moment where there is no effective change. So in an isotonic solution, iso is a prefix meaning the same. There is the same amount of water particles outside the cell as inside the cell. Essentially, there's the same amount of water particles outside the cell than inside the cell. So because the grass cannot be greener on the other side, there is no net water flow. Water essentially does not move across the cell membrane of the red blood cell. For this reason, this is an isotonic solution, and an isotonic IV is what will help make sure the red blood cells don't change shape, that it renders the red blood cell useless. In scenario A, figure A, we have a hypotonic solution. Recall that tonicity follows solute. Osmosis follows the solvent, typically water. And there is an inverse relationship between solute and solvent. So by extension, there is an inverse relationship between tonicity and osmolarity. In a hypotonic solution, the tonicity refers to the state of the extracellular fluid, what's happening outside of the cell. So if a hypotonic intravenous fluid or hypotonic fluid was infused, then the outside of the cell will have less solute than inside of the cell. What I'm really saying is that there is less water molecules inside the cell to begin with than outside the cell. And because there's more water molecules outside the cell, osmosis dictates that water will move from areas of high concentration, that's outside the cell, towards areas of low concentration, that is within the cell. So the net result of osmosis from when a red blood cell is basking in a hypotonic solution is cell swelling. There was a net flow of water molecules from an area of high concentration outside the cell to an area of low concentration that was inside the cell. And for this reason, the cell swells up, and we are concerned about this because it could very much damage the cell. In the other scenario, in a hypertonic solution, there is a lot of solutes outside the cell than inside the cell. What I'm also saying is that there is more water, more solvent inside the cell than there is outside the cell. So osmosis will dictate that water will diffuse from areas of high concentration to areas of low concentration. So the net movement of water is from inside the cell towards the outside of the cell, resulting in cell shrinking, also known as crenation. To summarize, we prefer an isotonic solution to be administered to our patients so that the shape of the red blood cell can remain a biconcave shape so that it can effectively deliver oxygen to different parts of the body. Water has a slight charge to it as an overall molecule. It is also not very large. So the main reason for why water can't pass through the phospholipid bilayer is because it's going to have a really hard time with the hydrophobic, the water-fearing areas of the plasma membrane. So in order to help water move from one side of the cell to the next, there are these transmembrane proteins that have a tunnel at the center that allows for movement of these water molecules accordingly. We are still following the concentration gradient, and so for this reason, this is still considered a form of diffusion, a form of passive transport. Passive mechanisms require no ATP. Facilitated diffusion is an example. Simple diffusion, which we talked about in reference to the respiratory gases, also are a form of passive mechanism. Things change when we start going against the concentration gradient. So recall the hill analogy I provided to you earlier on today. Here, we are moving up the concentration gradient from moving to from an area of low concentration to an area of high concentration. We need energy. We need energy in the form of ATP. And so the best way to identify whether a transport mechanism is a form of active transport or a form of passive transport, any active mechanism will require energy input. An example of this mechanism is the sodium-potassium pump. Here, we move sodium and potassium against their concentration gradient. In brackets, you have the name ATPase. And ATPase is an enzyme that breaks down ATP, as denoted by this notation here. The ATPase will break down ATP so that energy can be used for sodium to go up the concentration gradient, and for potassium to move up its concentration gradient as well. Sodium-potassium pumps are used in several scenarios throughout the body. This is one that I would keep in mind because it will show up over and over again. So in week 3, we covered concepts of cellular function. Now we are going to hone in on the cell division process, mitosis, meiosis, before we move into a discussion on tissues. So week 4 is entitled Genes and Cellular Function. It is worth revisiting cell theory, most especially the second last bullet point here, and that second cells will only come from pre-existing cells. This is what will help us understand the cell cycle. So the cell cycle essentially summarizes the lifespan of a cell. There are 2 main phases. There is interphase, which is comprised of 3 subphases, represented by a green arrow here. And then there is the mitotic phase, which is also part of the cell cycle. The cell will spend most of its time in interphase. You can see how the green arrow essentially takes 3/4 of the circle's perimeter. Remember that the M phase, which stands for the mitotic phase, is a process of cell division. The cell cannot afford to be dividing quickly or faster than it can handle. And so the cell cannot be in the mitotic phase too frequently or for long periods at a time. We are now going to break down the subphases. We're going to break down and address the subphases in interphase and then the mitotic phase. Interphase is comprised of 2 G phases. They stand for first gap, that's G1, and second gap stands for G2. Here, a lot of growth occurs. The G1 and G2 phases are separated by the S phase, also known as the synthesis phase. This is the time when cells duplicate their DNA information. This becomes really important because the product of mitosis are 2 daughter cells that are identical to one another, as well as identical to the parent cell that it came from. In the mitotic phase, there are 4 main subphases that we follow movement of the nucleus and chromosomes to determine what phase we are in mitosis, followed by a fifth subphase that has less to do with the moving chromosomes and a lot to do with the ideally even distribution of cytoplasmic contents within the cell. The best way, in my opinion, to memorize or familiarize myself with the process of mitosis is the acronym PMAT. P for prophase, M for metaphase, A for anaphase, and T for telophase. On the right side are microscopic images of what is happening at each of these four phases. On the left side are schematic, artistic drawings to help us understand what is going on. In prophase, the nuclear envelope starts breaking down. This allows for movement of the genetic material to different parts of the cell. This happens very fast, and so by the time the nuclear envelope is broken down, it starts to assemble again. And so effectively, the genetic material did not stay in the cytoplasm for long periods of time. The centrioles are there to help make sure each developing daughter cell has the right number of chromosomes. So metaphase is marked by the chromosomes lining at the equator at the center of the cell, the center of the cell. These centrioles between prophase and metaphase will start moving to opposite poles, arbitrarily selected, of the cell. And then it acts like a fishing line. It extends a protein line to the genetic material that's at the center of the cell and starts pulling it in like it's fishing. This ensures that each daughter cell receives the same amount as each other, as well as the same amount that came from the parent cell. Anaphase is marked by the separation of the sister chromatids so that each developing daughter cell has the exact same copy of genetic material. Telophase is marked by the chromatids reaching the opposite ends of their cell, of their new cell, and the nuclear envelope starting to redevelop. When we teach mitosis, we teach it in a linear format, but please understand that this is a very dynamic process. Before telophase even ends, cytokinesis has begun. And in prophase, metaphase, anaphase, telophase, PMAT, we follow the movement of the chromosomes. In cytokinesis, we do not. What we follow instead is the movement of the cytoplasm, ideally in an equal 50-50 distribution. It starts with a cleavage furrow in between the two developing daughter cells that pinches off completely, that to allow for ideally equal distribution of what each developing cell's contents, so that you have two daughter cells that look exactly alike and look exactly like the parent cell it came from. Mitosis will occur in all cells of the body except for the gonads, testes in males, ovarian in female. There are two key genetic terms that are important to know here. These are the words haploid and diploid. One of the tenets of cell theory is that all cells of the same species will look alike. One of these characteristics is the amount of DNA information that's found within the nucleus. Because we are a human anatomy and physiology class, all of the cells that we work with should have 46 chromosomes. You need 46 chromosomes, technically, to be considered human. 46 chromosomes for a total of 23 pairs. 23 is the haploid number. 46 is the diploid number. There are 23 pairs because these chromosomes look alike. Let's look at chromosome pair number 1. One of these came from mom, and one of these came from dad. And so the gonads, testes, where sperm is made, and ovaries, where a mature egg is released once a month, ideally during ovulation in the childbearing female. These gametes will only have 23 chromosomes because a worthy sperm needs to meet the egg that was released during ovulation to begin life, to start to make a fertilized egg. And after fertilization, there is rapid division right away through the process known as mitosis, as we discussed earlier in this presentation. So to summarize, the diploid count is 46 and the haploid count is 23. If all these cells of the body, except for the gonads, cells from the gonads and ovaries, go through mitosis, then meiosis is the process of cell division that occurs only in the gonads. What is unique about meiosis is that it requires 2 rounds of prophase, metaphase, anaphase, and telophase. 1 round of PMAT occurs in meiosis 1, and the second round occurs in meiosis 2. Meiosis can produce up to 4 daughter cells, 1, 2, 3, 4, each with 23 chromosomes, a haploid count. Contrast that with the 2 daughter cells that were produced as a result of mitosis. Those daughter cells also have 46 chromosomes, a diploid count. Now, what is unique about meiosis is an event called crossing over, which allows for genetic variability. Consider 3 siblings. I'll take my family as an example. I have 2 younger brothers. We all share the same parents, but we don't all look exactly alike. This is because the mature egg or sperm that were contributed from mom and dad to conceive each of us didn't have the exact same information as what was received from mom and dad to conceive both my brothers and myself. So the arrangement of 23 chromosomes can vary significantly, and this is due to a crossing over event that occurs in meiosis 1. There is no crossing over in mitosis. That concludes concepts from week 4. We will now explore different human tissues. There are 4 major tissue types. They are listed for you on this slide: epithelial, connective, nervous, and muscular. A tissue is defined as a group of similar cells working together to form a specific role in an organ. And this is a great time to revisit the levels of hierarchy, the levels of organization of the body. This is what begins the microscopic realm. They are similar cells, each tissue, and so all the cells within epithelial tissue will look similar as one another. They will look different from that of connective tissue and nervous tissue and muscular tissue. Because form follows function and function follows form, there are differences in the cell shapes as well as function of the cells found among the 4 major tissue types. Generally, what I like to do is to discuss the epithelial tissue, followed by nervous tissue, followed by muscular tissue, and then by default, anything that does not fall under the epithelium, nervous, and muscle tissue category will be considered connective tissue. Connective tissue is the catch-all of many, and so there is a lot of variety in connective tissue. While they will appear differently on histological slides, all connective tissue will have the presence of a matrix. The matrix is an extracellular material comprised of fibrous protein and a clear gel known as the ground substance. What we will do now is explore each of the major tissue types. The goal here is to identify key differences, look for hallmark characteristics that are key to each of these major tissue types, and be able to recognize and identify certain microscopic images should you be presented with them on the upcoming celebration of learning. Like mentioned earlier before, we are going to start with epithelium. Epithelium is a tissue type that's designed to line and secrete a substance. Because of its lining function, they are tightly packed cells. There is not much space for particles, matter, to pass through this lining. We describe epithelium based on whether or not there is one or more than one layer, so how many layers there are in the epithelium. And then we describe epithelium based on cell morphology, cell shape. So there are 2 questions we need to ask ourselves. One has to do with number of layers. Is there one layer? As in this example here, one layer of cells? If the answer is yes, this is a form of simple epithelium. If there is more than one layer, 2 or more, you do not need to count. That is referred to as stratified epithelium. In pseudostratified epithelium, pseudo meaning fake, meaning looks like, but not really. In pseudostratified epithelium, it's neither simple or stratified. There are several layers, but it is unclear. It is not a clearly defined layer. And so What can be really helpful here is to look at the nucleus, because under a microscope, the nucleus typically stains clearly, and the ratio is often one nucleus for one cell. And so if it is difficult to locate the cell membrane, then we can approximate the presence of a cell by the presence of its nucleus. The second question we ask ourselves is, what type of cell shape? There are only three options here. They are squamous, of a squished appearance, cuboidal, looks like a square, 4 equal sides, which we do not need to take a ruler out and measure, because the only other option remaining is a rectangular, columnar cell shape. Looks like a rectangle. So two pairs of sides, and it typically sits on the side with the shorter width. Squamous, cuboidal, and columnar. What proceeds are several slides, histological slides of epithelium underneath a microscope. This is simple squamous epithelium, one layer of squish cells lining the tissue. This is simple cuboidal epithelium, one layer of square-shaped cells that are tightly packed, lining the duct of a cell, or a tissue. This is simple columnar epithelium, one layer of columnar cells. This is pseudostratified epithelium. Here, it is unclear where the multiple layers are. The nuclei are also not layered clearly. This is best defined as pseudostratified columnar epithelium. By nomenclature, the layer at the apical surface of the cell that's open to the external environment, will take precedence with respect to the naming of the epithelium based on cell shape. All epithelium will have an apical surface and a basal surface. An apical surface, because it is a lining, there will be one side of the tissue that faces open space. And then the other side of the tissue that will be anchored to a basement membrane. This would be the basal surface. This is keratinized stratified squamous epithelium, which we will discuss more in the integumentary system in week 5. There are many layers of cells here that makes this stratified. Cell shape is squamous. And the keratinized structure is a protein structure located here. Contrast that with non-keratinized stratified squamous epithelium. So here we still have many layers of cells that are of squamous morphology, squished appearance, but there is no keratinized structure. This is non-keratinized stratified squamous epithelium. This is stratified cuboidal, more than one layer, square-like appearance of cells. This is often what's seen around ducts. My biggest tip for students when it comes to identifying slides underneath the microscope is what you see is what you get. When we start to misconstrue or push or start classifying structures so that they fit nicely into a box, that is often what leads us to mislabel or misidentify the epithelium at hand. So in this example, it is neither simple or stratified, nor is it clearly squamous, cuboidal, or columnar. This is best described as transitional epithelium, also referred to as urothelium, because this is what we see in the urinary bladder and the ureter, which are part of the urinary system. I mentioned two key functions of the epithelium. One was lining, and the other is secretion. The production and secretion of certain substances. There are two types of glands, because glands require or often involve the secretion of a substance. They are often packed with epithelium tissue. There is a difference between endocrine and exocrine glands. Endocrine glands typically involve the production and secretion of a hormone, and hormones are chemical messengers that have to travel to a very far part of the body to communicate. Because of this, hormones typically travel in the circulatory system, which is the highway of the body. It is the fastest way to get from head to toe if you are a protein. So all endocrine organs will have access to a blood vessel. Exocrine glands do not. Examples of exocrine glands include sweat glands and salivary glands. Sweat glands are found in the integumentary system, which is part of the skin. They produce a fluid known as sweat, which goes to the surface of the skin. So no hormones are produced by the exocrine glands that are the sweat glands. The sweat does not have to travel very far, and for this reason, does not need access to the bloodstream. What it will need access to is a duct. Make sure the fluid goes to exactly where it needs to go. The salivary glands are also an example of exocrine gland. Salivary glands produce saliva, and saliva will be deposited in the oral cavity, which is not very far from the salivary glands. There are three types of exocrine secretion listed for you on the slide. Eccrine, also known as merocrine, apocrine secretion, and holocrine secretion. The difference here will be in the amount of sweat, saliva, or whatever fluid this exocrine gland will be secreting. If only a little bit of substance needs to be secreted, then vesicles will form to bring the fluid to the surface of the cell, surface of the tissue, the epithelial tissue, so that it can be released. If a moderate amount of this substance needs to be secreted, then vesicles won't get the job done. It wouldn't be the most efficient way. And so if a moderate amount of secretion needs to take place, then a lipid droplet will form around the substance that needs to be secreted. And then if a lot of substance needs to be secreted, then the cell essentially sacrifices itself. It disintegrates, it dies in order to release this fluid. This is holocrine secretion. Eccrine, apocrine, holocrine secretion only apply to exocrine secretions. And if you are a visual learner, this slide is for you. The second major tissue type is nerve tissue, also referred to as nervous tissue. What's unique about nerve tissue is these cells have a stellate shape. Stellate, as in a star-like shape, as in stellar, the stars. The basic unit of nerve tissue are neurons. These are the actual nerve cells. And so in the neurons are where the electrical signal, the electrochemical signal that drives the nerve signal, these only pass through these neurons. These nerve signals travel very quickly, much more quickly than hormone signaling. And there are three regions to the nerve tissue. First, I look for the cell body, also known as the cell soma. Best way to find it is to look for the nucleus. Here is the nucleus for this neuron. The entire region surrounding it is the cell body. The extensions that are found around the cell body, these are short extensions, are called the dendrites. The dendrites receive the signal from the neuron that came before it. And then there will be one very long extension called the axon. The axon will deliver the signal from this neuron to the next neuron in an effort for the nervous system to send responses and signals very quickly throughout the nervous system. There are other cells that are found that are part of the nervous system, but are not neurons. So while these cells are not involved in carrying the electrochemical signals that is a nerve signal, they are called neuroglia, also known as neuroglial cells. They help protect and assist the neurons. They help support the neurons. They will do things like make myelin sheath. They will provide nutrition for the neurons. They essentially will, they're basically the neurons' support team to help them stay strong and help them stay efficient. We are going to circle back to connective tissue. I want to address muscle tissue first. There are three types of muscle tissue. There are smooth muscle tissue, there's cardiac muscle tissue, and there is skeletal muscle tissue. You may want to spend some time reviewing the differences, at least from a histology slide perspective. There is a system, if you will, that I recommend we take, and that is to ask ourselves two questions. Does the skeletal muscle, does the muscle in question have striations? And two, do they have intercalated discs? Before we address those, these slides are a little disorganized here. So the three types of muscle are listed at the bottom of this slide. Muscles will excite and contract. They are the only major tissue type that is capable of contraction in the human body. So there are two types of involuntary muscle. These are cardiac muscle and smooth muscle. Smooth muscle will line the organs, the internal organs. They help move things along as they should without us having to put too much energy into it, too much intentional thought into it. Cardiac muscle tissue is found in the heart. That's why it's called cardiac muscle tissue. When we don't think about beating our heart every day, nor do we think about contracting the esophagus so that whenever we eat, we make sure food moves down from our oral cavity, from our mouth to the stomach, which is below the diaphragm, inferior to the diaphragm in the abdominal pelvic cavity. Having to think about or putting intentional thought into contractions of the esophagus because of that's where smooth muscles are found every time we eat, or having to put intentional thought into making sure the cardiac muscles contract and relax at a decent rate would be so exhausting, you can probably imagine. So both cardiac and smooth muscle are examples of involuntary function, involuntary muscles. They don't require an external stimulus for them to work. Skeletal muscle does. Skeletal muscle are essentially the muscles that we try and work out when we go to the gym. And these are very much voluntary muscle because it takes work to achieve a lean body and to maintain a lean body. You can also imagine how much easier it would be if skeletal muscle were involuntary. We'd all be so fit all the time. How wonderful would that be? Now that we've identified the differences between skeletal, cardiac, and smooth muscle, at least with respect to function, let's talk about how they look different underneath a microscope. So, first question would be, does the muscle have striations? Striations are these appearance of track marks at the background of the slide, They run from top to bottom of the picture, top to bottom of the slide. Skeletal muscles have these, but also cardiac muscles. If you zoom in, you will see them. Well, who does not have striations? Well, smooth muscle. There are no tire track marks at all in these images. For this reason, the absence of striations creates a smooth texture. So this is smooth muscle. If the answer to your question of are there striations is no, answer smooth muscle and move on. If the answer is yes, you're not quite done yet because you have to decide between cardiac muscle and skeletal muscle. The difference between cardiac muscle and skeletal muscle are the presence of intercalated discs. Intercalated discs will appear as darker purple lines interspersed throughout the slide. You have to see several of them along the slide. They will typically run in the same parallel to the striations, but these will be more apparent because they will be thicker and darker. Intercalated discs mark an area of gap junctions. And why this is important is because when the heart contracts, all the cardiac muscle cells need to be able to contract at the same time. And so these gap junctions allow for these cardiac muscle cells to communicate with their neighbors so that when one of these cardiac muscle cells receives a nerve signal to contract, it can call on its neighbors, who will then call on their neighbors, and every cardiac muscle cell, ideally in the heart, will contract at the same time. This is what will provide the ejection fraction, the force by which blood will effectively move from the heart to different parts of the body to deliver oxygen to the cells that are there. If the cardiac muscle cells are unable to contract at the same time, the heart is essentially failing. And this is one of the things that's happening in congestive heart failure or other types of heart failure for that matter. So only cardiac muscle cells will have intercalated discs. Skeletal muscle cells will not. If the question is nothing to do with epithelium because there's no apical or basal layer, this tissue in question does not line anything, it doesn't secrete anything, or it's not a neuron, it doesn't have the ability to excite, or it's not any of the muscle subtypes we just talked about because it doesn't have the ability to excite or contract, then the best answer is connective tissue. Connective tissue will, there's a wide variety. Bones fall under this category. Cartilage fall under this category. Blood fall under this category. Tendons and ligaments also fall under this category. Tendons and ligaments are both comprised of the same type of connective tissue. They just attach different structures. And this is really important to note. Tendons will attach muscle to bone, whereas ligaments will attach bone to bone. So ligaments are typically found inside a joint, whereas tendons are found outside the joint. Because connective tissue, a lot of tissues fall under the connective tissue category. Connective tissue overall has several functions. They are involved in providing structural supports, protection, immune support, as well as insulation in the form of storage of certain electrolytes, as well as regulating body temperature because of brown fat, which is found typically in infants. It's one of the reasons that explain why infants tend to run hot, but because blood is a connective tissue, it carries heat, and the proper direction of or movement of blood in the body will help maintain homeostasis. Regardless of what connective tissue subtype is in question, what is inherent and what all connective tissue subtypes will share in common is the presence of a matrix. Only connective tissue will have a matrix. And this concludes my review for the upcoming Celebration of Learning number 2, more formally known as exam number 2. I hope you found this video very helpful. If you have any questions, don't hesitate to reach out via email or come by during office hours, and I am more than happy to discuss further. Happy studying.